Hello, students. Today we are going to explore one of the most exciting topics in biology, genetics. Why is it called the blueprint of life? Because everything from your height to your hair color is controlled by tiny instructions hidden in your DNA. Let's begin from the very foundation, the experiments of a monk named Gregor Mendel. Before Mendel, people believed that children were just a smooth mix of both parents, like mixing red and white paint to get pink. But Mendel showed that traits are passed down in a more organized way, in separate, tiny particles. We now call those particles genes. Ma'am, was Mendel a scientist? Good question. Actually, he was a monk and a teacher of science. He didn't even know about DNA, but his work laid the foundation for everything we know in modern genetics. Mendel chose pea plants, Pisum sativum, for some very smart reasons. They had clearly visible traits like tall or dwarf, green or yellow seeds. He could easily control their pollination. And most importantly, he used a large number of plants and applied mathematics. Imagine over 30,000 plants just for one experiment. Now let's understand Mendel's first law, the law of dominance. If we cross a tall plant with a dwarf one, all the offspring in the first generation, F1, are tall. Why? Because the tall gene is dominant and masks the dwarf one. So in a pair of genes, or alleles, the dominant one always shows up in the F1 generation. Now the second law, law of segregation. Let's say both parents are hybrids, TT cross TT. Their children can be TT, TT, or TT. If we use a Punnett square, the result is 1 TT, 2 TT, 3 TT. So, 3 are tall and 1 is dwarf. That's a 3:1 phenotypic ratio. Ma'am, how do we know the chances of getting a particular trait? We use probability rules. For example, the chance of getting TT is equal to 0.5 into 0.5 is equal to 0.25. That's the product rule. His third law is called the law of independent assortment. If we look at two traits, say seed shape and seed color, round versus wrinkled and yellow versus green, the traits separate independently. This leads to the famous 9:3:3:1 ratio in a dihybrid cross. That means 9 round yellow, 3 round green, 3 wrinkled yellow, 1 wrinkled green. It's like doing two monohybrid crosses at the same time. Now students, real life is not always that simple. Some traits don't follow Mendel's rules exactly. Let me give you some examples. In incomplete dominance, a red and a white flower give a pink one. In codominance, both traits appear, like in a blood group. Multiple alleles, more than two options. Blood group has IA, IB and I pleiotropy, one gene affects many traits. For example, in PKU disorder. Ma'am, can these traits be predicted like Mendel's? So far, we've talked about traits, but what exactly is a gene? A gene is a sequence of DNA that codes for a protein or RNA. In eukaryotes, genes have coding parts called exons and non-coding parts called introns. During processing, the introns are removed through splicing to form mRNA. Let's talk about the central dogma, the flow of genetic information. DNA, RNA, protein 1. Replication. DNA makes a copy of itself using enzymes like helicase and DNA polymerase. 2. Transcription. DNA is read to make mRNA. 3. Translation. mRNA is read by the ribosome. tRNA brings amino acids and proteins are made. Ma'am, what is the role of rRNA? rRNA is part of the ribosome. It helps build proteins and even acts as an enzyme. Now let's look at the amazing tools we use today, PCR, polymerase chain reaction, makes millions of DNA copies. Gel electrophoresis separates DNA fragments by size. DNA fingerprinting used in forensics based on STR's CRISPR-Cas9 gene editing scissors. Scientists can now remove, add, or modify genes in living organisms. Ma'am, is CRISPR being used in India? Sir, is CRISPR being used in India? Yes, in research labs and clinical trials. But it's still being carefully regulated for ethical use. So, students, genetics is not just about what traits we inherit. It's helping us fight diseases, understand our history, grow better crops, and even solve crimes. We've come a long way from Mendel's pea plants, and the journey is just beginning. That's all for today. In the next session, we'll practice some inheritance based problems and pedigree charts.